And if you're joining us from the previous session, welcome back. My name is Tom Ulmansky, and I'm a research scientist here at ISB, and I'm excited to chair the second session of our symposium, Natural History, Identifying Signatures of Within-Host Adaptation. Just a reminder that as the speakers present their slides, if you have questions, ask them directly via Zoom in the Q&A window, and please specify which speaker the question is addressed to. So we have two really fantastic speakers today, Dr. Nandita Garud and Dr. Mathieu Grosson. Dr. Garud will present first, and Dr. Garud, if you could start sharing your slides, that would be great, thank you. So Dr. Garud is currently an assistant professor in the Ecology and Evolutionary Biology Department at University of California, Los Angeles. Her research interests lie primarily in understanding the evolutionary dynamics of natural populations, including human microbial communities like the gut microbiome and the microbiome of Drosophila melanogaster. Her presentation today is titled Evolutionary and Ecological Dynamics in the Human Gut Microbiome from Birth through Adulthood. Please join me in welcoming Dr. Garud virtually. Take it away, Dr. Garud.
Thank you very much for this opportunity to present today. It's really a big honor to be amongst this group of people. So our microbiomes, as you know, play a very crucial role for our health. And we are seeded with one right from the moment we are born. So how do our microbiomes change over lifetimes? When I talk about changes in the microbiome, what I mean are ecological and evolutionary changes. For example, you might have species fluctuations over time, which are a regular feature of healthy microbiomes, but could also be an important feature of disease microbiomes that experience large fluctuations. And here I'm depicting species fluctuations by a shift in the colors of these circles that represent different species. Another type of change could be invasions of strains at the subspecies level. And so you might have different genetically distinct strains that come in and invade a person's microbiome, as depicted here with this X. And then you might also have evolution, in which you have a genetic change that arises on the background of an existing lineage that then rises to high frequency in its subpopulation, as depicted with this bullet going to high frequency in the red population. And when talking about changes in the microbiome over lifetimes, people have looked at many differences between infant and adult microbiomes. So for example, in one of my favorite papers by Backhead et al. In 2015, moms and babies over the first year of their life were sampled. And what was noticed is that there are different species compositions between moms and babies. And there are also different functional profiles. But we're really interested in asking the question of what about different evolutionary dynamics? Are the gut microbes in mom evolving differently from that of babies? And the reason we're interested in this question is babies have very different environments compared to moms. And I should say more broadly, adults actually, not moms particularly, I mean adults. And when I talk about these differences, what I mean are differences in diet. So in the initial months, the diet is very simple, comprising mostly milk. They have developing immune systems. They have simpler communities as well, which also might play an important role. And combined, all these factors could actually change the tempo and mode of evolution by changing the rate of adaptation, changing how adaptation occurs, either by de novo mutations or recombination seeded sweeps, and so on. It's important to quantify these differences because evolution ultimately is what generates new genetic variation in a population, which can then ultimately provide the basis for new traits in the population. So today, what I'm going to be speaking about is as follows. First, I'll be focusing on the infant, where we look at volatile ecological and evolutionary dynamics in infancy. Then we look at the adult, where we look at strain stability in the adult gut microbiome. And then we'll be looking at what happens over our lifetimes. And we see that strain loss and turnover seems to be inevitable. So first, I'll talk about the infancy. And this work is actually led by a very talented undergraduate in my lab, Daisy Chen, who is now a first-year PhD student at UC San Diego. When we started to work together, we asked the question of, are the evolutionary dynamics of gut microbiota different in infants versus adults? A lot of papers have been written in the past couple of years on evolution in the human gut microbiome. And these provide some very interesting and exciting insights into the field as to how commensal gut microbes could be changing. These papers have explored these very important topics using a variety of statistics and technologies. For example, some papers have looked at isolate data, and other papers have looked at Hi-C data. I would like to highlight the paper that I've been a part of, which is co-first authored with Benjamin Good, and also authored with Oscar Halachik and Catherine Pollard. Ben just spoke in the previous session and gave some highlights about this paper. And in this paper, what we found is that healthy gut microbiota in healthy hosts can evolve on 6-month timescales. And moreover, the rest of these papers seem to show that this is a regular occurrence of our microbiome. So Daisy and I, when we started to work together, we took the same approach that we did in the paper with Ben on analyzing shotgun metagenomic data to look for evidence of evolution. And we did so using publicly available shotgun data for more than 700 infants. These data come from 4 studies, including Backhead et al., Ferretti et al., Yassir et al., Shao et al., and also we included the Human Microbiome Project to have a comparison with healthy adults. What you can see is that the distribution of ages span birth through the first year, and the first days and weeks are actually very densely sampled, then every month thereafter. And many of these samples are paired with mom. And so that allows us to look at how bacteria change once they are transmitted from mom to infant. So briefly, I'll go over some of the technical details from our previous paper so that you can understand how we arrived at our conclusions looking at infants. And so 16S data is very useful type of data for looking at species fluctuations, but not so useful for looking at invasions and evolution because it doesn't give you information about the rest of the genome. And so instead, we can use whole genome shotgun sequencing data to look at this type of processes. And so how do we detect evolution with shotgun reads? Ideally, we want to look for mutations that go from low to high frequency over time. But as you can see, this is a very messy type of data with a lot of species all mushed together. And so we want to simplify it by looking at within species genetic variation. And so what we do is we use a reference-based approach where we map reads to individual genomes, and then we can look at allele frequencies at individual sites within each species. And this tool that we use is called MIDAS and comes from Katie Pollard's lab. And in doing this, we can then look for genetic differences between samples. So for example, here's a panel of hosts, and you can see in this blue species that you have a mutation present at 0.8 in this host, 0.3 in this host, 0, and etc. And this allows you to look at the genetic variation across hosts and draw some interesting population genetic inferences. It also allows you to track strain transmissions. So for example, in previous work, we looked at transmissions of strains from moms to infants by looking at private variants that are present in mom and infant only and absent in the broader population. And using this technique, what we found is that the number of shared species increases over time, but marker allele sharing decreases over time, indicating that even though the microbiome of an infant is maturing, many other sources besides mom may be playing an important role in seeding the baby. Now, detecting evolutionary events is not as simple. And I should say that while this type of statistical signal is really useful for strain tracking, it doesn't really give us the resolution to look for evolution. because really what you need to know are the haplotype backgrounds that some of these mutations are rising on, so that we can distinguish them from sampling error and also strain fluctuations, which I'll get into right now. So ideally, what we would want to do when we look at this type of data is look for mutations that go from low to high frequency over time, indicative of evolution. But the complication is you might have multiple strains colonizing a host at high frequency, as shown here with the red and the blue strains. And these strains could be highly diverged. So here I show a strain of, let's say, B. vulgaris in this host and in this host. And the evolutionary time separating these two strains could be sufficiently great that you have a lot of divergences accruing. By contrast, when we want to look for evolution, we want to look for a small handful of differences that accrue on short timescales over a few months. And so these large differences will manifest themselves as polymorphisms in the sample that then can result in allele frequency changes just due to strain fluctuations. So we want to resolve that lineage structure to be able to detect evolution. And the tricky thing is that all we witness are allele frequencies. So somehow we have to go from these allele frequencies to inferring this underlying strain structure. How do we do that? So in this work with Benjamin Good, what we did is we plotted the distribution of allele frequencies such as 0.15, and you can imagine we're taking a bunch of frequencies throughout the genome, and we plotted this distribution. So this is for the species Bacteroides vulgaris, and we're showing data for 3 hosts from the Human Microbiome Project. On the x-axis is the allele frequency, on the y-axis is the fraction of sites. And what you see is that there's a lot of variation across hosts in their distributions. Now, these patterns are actually inconsistent with a single strain colonizing a host and then diversifying. In such a case, you'd expect a bunch of sites to have very low frequencies arising from the diversification of the lineage within the host. But instead, we see these bumps at intermediate frequency, and these are actually indicative of multiple strains colonizing the host. So here, for example, you have a strain at 15% and 85%, and the divergences between these strains manifest themselves as polymorphisms at this intermediate site. Here you have the same pattern, but shifted to 50%. And here you have multiple strains, maybe 3 or 4 or some discrete number that are generating multiple bumps. Our goal here is to confidently infer a haplotype from the sample so that we can assign mutations to that haplotype to distinguish evolution from strain invasions. So we exclude samples that have sufficiently complex lineage structure so that we can instead focus on samples that are simple enough to confidently phase. And so here, for example, if we take a look at pileup of sites between 2 sites, on average, you'll have a site at low frequency, an allele at low frequency, let's say 15%, and one at higher frequency at, let's say, 85%, corresponding to the relative frequencies of your strains. And you can see that you can assign the dominant nucleotide to the dominant strain, thereby confidently phasing it. But not all samples are so amenable to this technique. In this kind of sample, you would have 2 strains at roughly 50-50 frequency, and you actually can misassign the allele with equal probability. And so that means that you cannot confidently phase a haplotype in this type of sample where there is no clear dominant strain. And so we only focus on those samples with really simple lineage structure, which fortunately there are a lot of in our data set. And in these types of samples, we can then look for mutations that go from low to high frequency over time. And this would be a signal of evolution. Okay, so we apply this technique to publicly available infant shotgun metagenomes, and I've shown you this figure before. And we looked, and in this temporal data, we asked how many sites go from low to high frequency over time per pair of confidently phasable samples. And Ben actually showed a very nice plot in his talk, but I'll recapitulate it here. Within host, when you're looking at two confidently phasable time points, you might have a wide range of SNP changes. So we observed actually a bimodal distribution in which you have either a small handful of SNP changes on the order of 5 or so, or you might have a very high number on the order of about 10,000. And if you look at the average, it comes out to be about a few hundred, and that's reflecting actually a mixture of these two very different numbers that are arising. And so this bimodal distribution actually reflects two processes. They reflect invasion of strains and also evolution of strains. And the evolution, again, should result in small numbers of SNP differences, whereas invasions should result in large numbers. And in fact, if you look at the average number of SNP differences between different hosts, you get on the order of 10,000. So by using the between host comparison, you can control for what's going on in terms of evolution versus strain replacement. Here we're calling it invasion instead of replacement. And so now that we can look for evolution with this public data, we then went ahead and looked at what the rates of SNP changes are early in life due to evolution. So looking at only those samples that experience either no SNP changes or evolutionary changes, we computed this rate per day by looking at the number of SNP changes between time points and dividing by the total amount of time. And we looked at different age categories. So we looked at the mom to infant transition. So basically, if you have the same strain that makes it from mom to infant and then acquires new genetic changes, then we looked at the first week of life. So two time points collected in that first week. We looked at week 1 to month 1 to year 1. And then this adult category are 2 adult time points collected from the same host from the Human Microbiome Project data. And what you see is actually that this rate seems to decay with life stage. And this was a very surprising result to us. And initially, from the mom to infant transition, we have a lot of SNP changes, or a very high rate of it. And then by the time you reach the first week, this rate seems to drop tenfold. And then by the time you reach adulthood, the rate really drops quite a bit. And you might be asking, we have different durations here. Could that be reflected or influencing these rates? We actually did a control where we matched the amount of time passing between 2 time points in adults versus 2 points in infants. And we see that the infants have statistically higher rates of SNP changes over time in the same time interval. So we did the same kind of analysis, but looking at strain replacements. And you can see that over time, this seems to decay. And so initially, replacements are really fast. I should note that this is between 2 QP samples. And so, there could actually be a lot more strain loss. But this is not actually, is not that surprising given that there's a lot of species turnover. And so naturally, there should also be a lot of strain turnover. Things are still volatile through the early parts of life, but then eventually settle out into adulthood. And this is actually a point that I'm going to return to in the second part of my talk, the fact that eventually our microbiomes in adulthood seem to stabilize and strain replacements become relatively rare. And then finally, we looked at gene gains and losses. And here, gains and losses can arise through horizontal gene transfer. Loss can also can arise through spontaneous mutation. And so what we see is some interesting patterns. So during the mother to infant transition, you can have more loss than gains. And this seems to concur with the fact that you lose a lot of diversity at the species level as well. But you also seem to lose a lot of diversity at the gene level. Later on in life, you see this recovery in diversity, and you see a lot more gains than losses. And this seems to be concurring with the fact that over time, microbiomes seem to functionally mature. And this gains could be a mechanism by which it acquires that functional maturation. Over time, eventually in adulthood, the rate of gains and losses seem to settle out, and they become roughly equivalent. But overall, the rates of gain and loss seem to be higher in infants compared to adults. So I've just shared with you some results by Daisy Chen that we recently put on bioRxiv, looking at the differences in the ecological and evolutionary dynamics between infants and adults. And so this brings me to the second part of my talk on strain stability in the adult gut microbiome. And so going back to this figure, strain replacements become less frequent in adulthood. And you can see that overall, the rate of strain replacements becomes very low. And so this was something that we noticed also in our previous paper with Benjamin Good, in which replacements generally seem rare in healthy adults on 6-month timescales. And so we asked the question of how stable are strains in adulthood in healthy hosts. This work is led by a very talented PhD student in my lab, Richard Wolff, as well as a very talented postdoctoral fellow in my lab, William Shoemaker, who's funded by the NSF. And we recently put a preprint online in which we asked about the ecological stability of strains in the human gut microbiome. And so previous work, such as by Schleswig et al. 2013, has shown that genetic diversity, variance in genetic diversity within a host over time is lower than between hosts, suggesting that hosts harbor a kind of a genetic fingerprint that persists with us at least on 1 or 2 year timescales. And so we took some data, or reanalyzed some data from Poirier and Grusin and Zhao and Lieberman, in which we looked at pi in the common gut commensal Bacteroides vulgaris over almost 2 years in 200 time points. And you can see that pi kind of fluctuates around some mean, but it generally seems to return back to that mean value. And we then, Ricky looked at the strain frequency trajectories, and he phased the strains using a strategy in Rudgar and Good et al., which was mentioned in Ben's talk. And what we found is that there are roughly 3 strains present. And you can see that their trajectories also seem to fluctuate over around some kind of constant value. They might deviate from that, but eventually return to it. Just like pi, you can see that where we have a perturbation in pi, we also see strains becoming more even in their distribution. And so this kind of provides a visual check of 2 different summary statistics of the same data. And like Jesse had mentioned in his talk, the strain inference there's another paper by Zheng et al. In which strains from the same person and the same species were inferred using microbe-seq droplet technology using barcodes, and they also find these similar trajectories, suggesting that our inference is robust. And so we asked the question of whether these fluctuations in strain frequencies are, what is the typical expected range of fluctuations in a healthy host. And so to ask that question, we took a page out of the literature, which had been asking the same question, except at the species level. Species also fluctuate in their abundance over time. And recent work in the past year or so have shown that macroecological laws can reasonably describe those species fluctuations. And in addition, a model called the Stochastic Logistic model can actually recapitulate fluctuations of species abundances over time. And so we asked whether these same kinds of models are applicable at the strain level. And so more details about what this Stochastic Logistic model is, it describes abundance fluctuations around a fixed carrying capacity. So here's a logistic model. You can see that there's some growth rate, and then there's a carrying capacity as illustrated with this dashed line. You can have some stochasticity that's described by environmental noise and Brownian noise. And Ricky simulated hundreds of times this model, and you can see these individual simulations here in light red. And you can see that there's a kind of a width to this distribution that kind of describes what the typical range of fluctuations are under this model that describes eventual stability. If you were to grab different points in this stationary part of this distribution, you actually will get a gamma distribution describing the frequencies of this strain. So we fit this model to data, and to do so, we need to know free parameters, just the mean and the variance and strain abundance. And this is what the fit looks like for at least 2 strains in the data set. So here is a strain for B. vulgatus, and here's a strain for Bacteroides fragilis. And we simulated this model and the SLM. And you can see that the red for these different simulations encompasses the trajectories observed in the data, suggesting that actually this model is a good fit for the data. Moreover, if you plot the distribution of these blue points, you can actually get back the gamma as mentioned earlier, suggesting that indeed this SLM is a good model for the data. And so we fit this model to strain abundance trajectories from 4 hosts from roughly 50 strains. And these are the 4 hosts in this Poirier, Grusin, Zhao, and Lieberman data set. In the vast majority of cases, roughly 95% across hosts, the SLM was sufficient to describe the dynamics. And what this suggested is that, in fact, strains in healthy hosts are stable and can actually be described by this model that describes fluctuations around this fixed carrying capacity. To further investigate this, we probed into what the typical size of these fluctuations are and whether macroecological laws can actually describe the statistical patterns observed across multiple strains. And so we plotted all the different strains here, each colored in a blue point. And we're looking at their mean abundance and their variance in abundance over time. And you can see that on a log-log plot, this is linear, so suggesting a power law. And this is consistent with Taylor's law, which is a macroecological law. And then we also plotted this, all the gamma distributions for each of the trajectories. And you can see that they generally can fit a similar distribution. So together, the fit of these 2 macroecological laws and the fit of the stochastic logistic model seem to suggest that, like species, strains are stable ecological units in the microbiome. And strains of the same species can coexist stably. And perhaps this is because they differ ecologically, or maybe because of spatial segregation along the gut. But there's an interesting caveat to this. So this brings me to the very last part of my talk, which is very brief, and that is strain loss seems to be inevitable over our lifetimes. And so this period of constancy doesn't seem to stay with us forever. Eventually, all strains are replaced over 20+ year timescales. So to arrive at this conclusion, in the paper with Ben Good in PLOS Bio, we looked at data from twins. And we leveraged a finding that was published by Cropello et al., which showed that cohabiting teenage twins and siblings share many strains. A data set by Shi et al. Had sampled 125 adult twin pairs that had lived together for 20 years and then lived apart for 40 years. And then eventually, we asked the question of, do these adult twins share the same strains? And the answer was no. And so that means that whatever you have in your guts today might turn over in the future, or rather it will turn over in the future. And so there seems to be limits to the stability of our gut microbiomes. And also, moreover, any adaptation that seems to be happening seems to have limits because eventually the strains are lost. Why they are lost, when they are lost, how they are lost, are all open questions for future research. And so to conclude, what I've talked about today are volatile ecological and evolutionary dynamics in infancy that seem to settle out into adulthood. We looked at strain stability in the adult gut microbiome, but found that over our lifetimes, strain loss and turnover is inevitable. I'd like to thank the many people who worked with me on this work. It has been a huge pleasure to work with all of them. They include Daisy Chen, Richard Wolff, William Shoemaker, Benjamin Good, Katie Pollard, and Oscar Halachek. I'd also like to thank my funding. And thank you to the organizers for giving me this opportunity to speak at this symposium.
Thank you so much, Dr. Garud. That was a really fascinating talk. We'll move on to Dr. Grossel's talk, and then we'll have a panel discussion at the end of the session.